All right, our next session is with Dr. Julia Maeve Bonner. Dr. Bonner has a PhD in molecular genetics from the University of Toronto, and she did her postdoc at MIT. She's now the principal scientist at Sanofi. Dr. Bonner, I got to tell you, out of all the amazing presentations that we saw at the New Directions in Choline Symposia, yours was my favorite. And I think that's probably because, it was a bit personal for me. I've got two grandmothers that passed away with Alzheimer's. My mother passed away a couple years ago with Alzheimer's. And I have a younger brother that's got the early onset version of that. So I was obviously very attentive in listening to your presentation, found it very interesting. If you wouldn't mind, kind of just give us a general overview of kind of the high points of what your presentation was about at the
Sure.
conference.
First, I'm sorry to hear about your grandmother and your mother and your brother. This is why we do this work. It's a devastating disease. So I'm sorry to hear about that. I hope that we can use some of this work to move the field forward. So the summary of my research is at MIT. So in the lab of Dr. Li Weicai, I was working on a genetic risk factor for Alzheimer's disease, ApoE4. So ApoE4 is the most validated, highly validated risk factor, genetic risk factor for Alzheimer's disease. Occurs in about 14% of the population, the control population, and is enriched to about 50% of Alzheimer's disease patients. And it is dose dependent. So, the more, if you get one copy of ApoE4, you have an increased risk of about 4-fold. If you have 2 copies, it's about 10 to 12-fold. So it's clearly very closely linked to Alzheimer's disease risk. And we wanted to understand, what is happening in ApoE4 compared to the non-risk or neutral risk allele, rather, which is ApoE3. What's happening in the risk version versus the non-risk that's changing the cellular state, changing the brain ultimately to create this sort of early system of risk. So we were characterizing this gene and this variant of the gene, the risk version, in the cell type in the brain that makes the most of this protein. So these are astrocytes. These are glia. They support neuronal cells. They also have many functions in the brain, one of which is to sort of it's a major modifier of lipid homeostasis. So this is the fats in the brain that are, the cell surface, the membranes of the brain, the intracellular organelle membranes, and, many important components that get transferred that are important for cell function and then get transferred between cells. And ApoE, the protein, is one of the major facilitators of transport of these kind of lipids, these fats between cells. And so what we found is that in this brain cell type, astrocytes, there was a really distinct shift in the lipidome. This is all the kinds of lipids that are being made or accumulated in the cell. And the ApoE4, the risk version, accumulated neutral lipids to a much higher degree, a particular kind of lipids, especially these triacylglycerols. These are the kind of lipids that accumulate in these intracellular compartments called lipid droplets. This accumulation is associated with dysfunction of many different kinds of functions in the cell. We found that using astrocytes, as well as from work that we started initially in Susan Lindquist's lab at the Whitehead in yeast, that we were able to pinpoint a node, a biochemical node, where we could kind of influence this lipid dyshomeostasis and the resulting outcome for the cell. So we found that we were able to discover that if these cells were grown in a media that was supplemented with choline or rich in choline, compared to media that was deficient or minimal in choline, that we could really alter this lipid imbalance and move it a lot. If in the media that was supplemented for choline, move these cells quite a bit closer to the cells that were in this non-risk state, the ApoE3. We could move that lipidome, so all the kinds of lipids it's making, but especially these neutral lipids that were accumulating, these triglycerols, we could move them a lot closer to sort of ApoE3 or non-risk levels. And then through a lot of genetic work, we were able to pinpoint that this was actually phosphatidylcholine synthesis. So we saw that the Kennedy pathway of phosphatidylcholine synthesis was critical for this effect of choline on modifying the lipid balance. We then have been, since then, that work was published in Science Translational Medicine. And since then, we've done a lot of work to try and see, could we move this into a potential, therapeutic in an in vivo in an intact animal. So we were looking at mice that have Alzheimer's disease genes knocked in. So these are amyloidogenic genes. This means that these mice have mutations, human genes with mutations in them that cause them to accumulate the plaques that we see in human brains in Alzheimer's disease. And also in the background of this, they had the human ApoE knocked in, which means that they're expressing human ApoE either 3 or 4. And in this ApoE4 background with this amyloidogenic mouse model, we fed them diet that was high or low in choline. And we saw a really nice protection against the accumulation of some of this Alzheimer's-related damage, as well as a shift in the lipidome of the cells that we looked at, and some of the tissues we looked at. And we're still characterizing these outcomes, but we're excited. We think that, it's possible that something that is as simple as a dietary supplement could maybe merely make a difference, especially for those who have a copy of this particular risk allele. So could be basically through modifying this lipid imbalance, we're hopefully kind of restoring a little bit of the norm or non-risk state to the cells that might otherwise kind of be in this imbalanced or risk state. So that's the gist.
Yep, that's a big picture. I'm kind of curious. So if and I'm going to ask you a question I know you don't know the answer to, but I'm going to ask you to speculate, so if you were to use choline supplementation to kind of stave off Alzheimer's, if you had the ApoE gene, would you have to be taking that or supplementing throughout lifetime? When would be the best time to be making sure you had enough choline supplementation in your diet?
So, we can't say these are mice and cells in a dish. We have human cells in a dish and they're mice. So we, of course, have to be very careful about making any claims that this, can have the beneficial effect in humans that we would hope. There have been previous clinical trials looking at choline supplementation in Alzheimer's disease with some mixed results. A lot of those weren't stratified for ApoE status, like whether they were ApoE4 versus ApoE3. And there is one study where it does look like when they did stratify that the ApoE4 responder or carriers were responded a little bit better. But we'd need to do a more careful, very on purpose, intentional study in humans looking at, different levels of choline with, in ApoE4 carriers, having tracked, ideally looking at sort of certain lipid markers maybe in their blood and following that, a clinical trial is required to be able to say this. And we need to be careful to make sure we don't overstate claims. But I think that, our science suggests that it should be possible and that it's worth doing these kind of studies to identify if there is, an intervention like a supplement, like food, like, eating choline-rich foods that could make a difference, because of course this would be a wonderful thing. One thing I can say is that in general, according to sort of nutritional studies, people tend not to get enough choline just by, Surgeon General recommendations. So I think that it's probably safe to say that you should get as much as is recommended currently, and then hopefully we can, do more studies to really identify if it has a specific benefit for APOE4 carriers.
And can you talk a little bit about maybe are there future research studies planned either by yourself or
So
others?
there is a small study currently being planned in collaboration with MIT and the Neurodegeneration Consortium out of MD Anderson, and I think with Balchem, that have sort of been working together to try and get not a full phase 1 clinical trial or phase 2, but a clinical study in humans to try and identify biomarkers that would change on different levels of choline, and then this would create the kind of readouts we would need to know before going into a clinical trial. But of course, clinical trials that are then very expensive, so we'd have to sort of see what happens after that. But this would be, hopefully a proof of principle that we can change important and relevant biomarkers, and then those can serve as readouts in a potentially bigger study.
Eric, you have any questions?
Yeah, I'm just curious if maybe you can comment on what the relationship is between, say, choline intake and neurodegeneration in epidemiological literature. Um, is there a connection between these two things? I mean,
So there's correlative evidence, which is that groups that are at risk for low choline, there's all an overlap of groups that are at risk for low choline versus groups that are at high risk for AD. So, for instance, women are at higher risk for low choline. Both from in the um, potential childbearing times when there's, because choline is also in the folate cycle, folate metabolism pathway. So uh, there's a much higher requirement for choline in women, and so then they are potentially at risk for being older people. It's been reported that the elderly, there's a decrease in their ability to uptake choline, even to whatever level that they have it circulating from their diet, that there's a decreased ability to take it up. So this could also increase risk, and of course, we know that age is the biggest risk factor for AD. And there are some studies that suggest that AD patients are, have lower levels of choline, but it's not super consistent. So that would, those studies, it kind of depends on what's being measured.
definitely an interesting emerging area for sure. Really neat to see sort of some of this pioneering work you've done mechanistically. I think the picture is very complex. I mean, as you said, this relationship between ApoE4 and other dietary factors is tricky, and I think something that's only been appreciated in the last, I don't know, let's say
Mm-hmm.
10 years or so, really to a certain degree. I mean, I do, the reason I love your work in particular, one of the reasons, is because um, I do think this appreciation for specific genetic markers and how specific dietary interventions might help people with those genetic markers, I think is incredibly important. We're definitely at a stage, I think, in the field of nutrition where um, a lot of the sort of big picture general population recommendations have definitely been covered. And I think this area of nutrigenetics, precision nutrition, personalized nutrition, whatever buzzword you want, is definitely the future. So
Yeah.
I really
Well, thank
enjoy
you. And I think
research.
I agree. I think that, similarly from, a field of Alzheimer's research, we've done a lot where we try and look at sort of a broad scope of Alzheimer's disease suffering patients and compare, all AD against all control. And, the more we learn, the more we understand that there might be subtypes within the disease. There might be, within that all kind of converge on a clinical presentation of memory defects and neuronal loss, but there might be underlying mechanisms that are slightly different or have slightly different sort of origins or starting points. And if we can identify that that's the case, if we understand whether or not that's the case, and then we can potentially understand that there are more targeted therapies that might be more amenable to some AD patients versus others, and that if we understand sort of how they're getting to their clinical presentation, we can have a more effective series of therapies.
In your animal study, do you know what the equivalency would be to a human diet? For example, in the supplemented mice, were they getting?
Yeah. So the choline dietary recommendations for mice are very high, much higher than for humans. We went basically our dietary paradigm. We wanted to kind of try and keep as within the scope of what sort of healthy mouse would see. So we went to sort of the National Research Council minimum and maximum, and those were about fivefold different between the minimum choline and the maximum choline, about fivefold different amounts of choline. But those are much higher. They're in grams per kg amounts of choline in the diet compared to 450 mgs per day that is recommended for adult males or adult females. 450 mgs per day, somewhere in that range. There are much higher requirement in mice, so we were within the sort of healthy mouse range, and then a fivefold increase in the supplemented diet.
I do think your comment is very prudent, right? I mean, I think what we know, of course, that before making any kind of strong claims about the connection between choline and the disease, of course, we need rigorous RCTs. But I do think as a prudent dietary piece of advice, it's probably smart to at least stick to trying to get your daily requirement. So I would agree. I think you're spot on with that recommendation. Just tough. Not a lot of people do that, right? I think you said depending on the source, it's what 6 to 10% of people get enough. I mean, it's really not a lot. So a lot of work to be done in terms of education and awareness.
That it's tough to get the appropriate dietary amount of choline. So humans do make some choline, but not enough. It is a required nutrient. So it can, it's depending on your diet. I think, the recommendations that are always the case for most diseases of aging, most healthy living in general, which is, a good balanced diet and taking care of yourself in every way that you're able to, are always safe recommendations. But again, in terms of specific recommendations for choline intake beyond the already, we would need to do the careful studies. But most people don't even achieve that much. So it seems prudent to watch what you're eating and do your best.
Dr. Bonner, as we get ready to close out here, any key takeaways you'd like to leave with our audience?
Well, I appreciate, that this is one of the sort of newer angles of looking at Alzheimer's disease is looking at it from a sort of metabolic, potentially nutritionally accessible viewpoint. I think one of the key, one of the things that will help, I think, to understand different kind of interventions and how they can be most helpful is these kind of mechanistic studies, these kind of this work where we can understand, there are broad benefits to the things that we know our body needs, but if we can actually, start linking those to specific pathways, to specific mechanisms, to specific, requirements that may be greater or lesser in certain risk states, then I think, there's a much greater case. It's much easier to convey that information to a general public, but also hopefully to convince, the medical field that these are valuable insights. And of course, with that, I just would also like to say that all this work is, it's publicly funded. We are excited to share it. We hope that people can learn from it. And, the most important thing is if we can try and make a difference for people who are suffering from this terrible disease.
Yeah, well said. I want to thank you for your work on this important subject, and I want to thank you for joining us here today.
Yeah, happy
Appreciate
to. Thank you.